Hello students, welcome to our third lesson in cell physiology, and we want to discuss osmosis. And osmosis is the process by which solvent molecules move from a region of high concentration to a region of low concentration through a semi-permeable membrane. So solvent molecules is just like water molecules, and they are moving from a region where we have high concentration of these molecules, and that that solution which is having high concentration of water molecules is a dilute solution. And then these water molecules is moving to a region of low concentration of water molecules, meaning this solution of low concentration of water molecule is highly concentrated with solute particles, and it must occur through a semi-permeable membrane. Osmosis can also be described as a special type of diffusion, since it involves the movement of solvent, that is water molecules, from its region of high concentration to a region of low concentration. So we have a practical activity, and we are demonstrating osmosis using a whisking tubing. We require beaker, a piece of whisking tubing, thread, glass rod, concentrated sugar solution or salt solution, and distilled water. So what are we supposed to do? We have to put 350 centimeters cubic of distilled water into the beaker, and then dip the whisking tubing into water to moisten it, and then we tie one end with a thread. Then with the open end, we have one end which is open, we pour the sugar solution to halfway and tie it. And during this procedure, we must ensure that there is no sugar solution spilling out of the tubing. And then what follows is that we immerse the tubing into the distilled water in the beaker and suspend using a glass rod. So you have to check after 30 minutes. So this is your setup. Have filled the tubing with sugar solution, and then you fill your beaker with distilled water, immerse the tied or the suspended whisking tubing on a glass rod. Then after 30 minutes, you come and observe. You are likely to see the following: that the whisking tubing became swollen, as you can see, indicating that its cell contents increased in volume, and the amount of water in the beaker reduced, implying that water moved from the beaker inside the whisking tubing. And you should know that, both the whisking tubing contains both water and sugar molecules, because you dissolve the sugar molecules to get the sugar solution. Then the beaker contains a higher concentration of water molecules than the whisking tubing. Therefore, water molecules diffused or moved from the beaker here, where there is higher concentration of water, into the whisking tubing where they are lowly concentrated, or there is low concentration of water molecules. And even though there is a higher concentration of sugar molecules in the whisking tubing, they were not able to diffuse or to move into the whisking tubing because of their large molecular sizes. So other than using the whisking tubing, we can use the dialysis tubing or cellophane, which are also semi-permeable membranes in this experiment. And the whisking tubing is commercial, synthetic, transparent, semi-permeable membrane, and it represents the cell membrane in the living cells. So how did this happen? We can now explain osmosis in detail, that when two separate solutions are separated by a semi-permeable membrane, there will be movement of water molecules or solvent molecules from their region of high concentration to a region of low concentration across a semi-permeable membrane. And this semi-permeable membrane will not allow movement of solute particles across it. So solute particles are just like the sugar or salt particles. So the movement of water molecules continues until the separate solutions have the same concentrations. We can also investigate osmosis in living tissue as our third practical activity, but the second in osmosis. So you have to have a potato, then push a cork borer through a large potato, and then you remove cylinder tissues from the borer just to get 4 cylinders of potato from. Then you chip or cut off the ends to have a 50mm length of cylinders, and just feel their texture. After having felt their texture, you place the 2 cylinders, the 2 out of 4, you place in a boiling tube containing distilled water, and the other 2 potato cylinders in the boiling tube containing a strong sugar solution. And then you just leave the setup for 30 minutes. After 30 minutes, you remove the cylinders and mop or dry with a tissue paper, and you have to measure and record the length of each cylinder. Then explain your differences. So what are your expected observations? This is an experiment you can try in your home. When you don't have a borer, just use a ruler to obtain or any other measuring instrument to obtain cylinders, or even if they are rectangular in shape, but they should be of equal length, not necessarily 50mm. Just try that in your home. You will see that the potato cylinder in distilled water became rigid, and those in sugar water became flaccid or less rigid. The reason is that if a plant cell is placed in water, that is distilled water or a hypotonic solution, the cell wall will draw water from the hypotonic solution. Hypotonic solution is a solution which is composed of mainly water particles or water molecules. So it is a lowly concentrated solution, meaning, or it is a dilute solution, meaning it contains more of water particles than the sugar particles. So the cells will obtain water from the hypotonic solution through osmosis, causing the cell to distend, and it becomes firm and rigid. See, plant cells is composed of cellulose, which is rigid and does not allow the plant cell to burst, as in the case of animal cell. So this water will just, the plant cell will just draw in water, become rigid, but it will never burst. Reason is because of the presence of the cellulose cell wall, which is rigid and firm, does not allow bursting. And when you place in hypertonic solution is the one which is containing more of the sugar particles and less water particles. The cell will lose, the plant cell now will lose water through to the solution, to the hypertonic solution through osmosis, and as water moves out of the cell starts to shrink, becomes less rigid or flabby, and such a cell is said to be flaccid. So solutions with the same concentrations are said to be isotonic. That is, if the solution of the cell and that of the surrounding is the same, then it is said to be isotonic. So those solutions are said to be isotonic to each other, meaning they have the same concentrations or the same volumes of either solutes or solvent. But then, so we can describe a lowly concentrated solution, that is a dilute solution, being referred to as hypotonic solution, and it has less of the solute particles, that is solute, I said there are particles like sugar or salt, and it has more of solvent particles, this is, or water particles. So a highly concentrated solution with more of the solvent particles but less of the solvent particles is referred to as hypertonic solution. So a highly concentrated solution will contain less of sugar or salt particles. All these are referred to as solute particles. And when isotonic solutions are separated with a semi-permeable membrane, there will be no net movement of solvent molecules to any of the solutions, since they have the same concentration of solvent particles. So those solutions are said to be isotonic to each other. So our next thing we want to look is what we call osmotic pressure. So when a concentrated solution is separated from distilled water by a semi-permeable membrane, then the concentrated solution will develop a force with which it draws water through the semi-permeable membrane from the distilled water. So osmotic pressure refers to the force with which a concentrated solution draws or gets water to itself. And an osmometer is used to measure osmotic pressure. Then apart from the osmotic pressure, we have the osmotic potential, and this is a measure of the pressure a solution would develop to draw water molecules from pure water when separated by a semi-permeable membrane. So we are done with that lesson, and just go through this assignment and discuss with your friend, and remember all the answers is just within the notes. So you are a great student and you are a great achiever. Have a good time.